Welcome everybody to the second in our webinar series. It's great to have you all here with us today. So thank you for joining. So just a few housekeeping. So we are going to be running polls throughout the presentation. So every now and again, you'll see one come up. The speakers will introduce them. And so you can then answer those questions. The other thing, we are recording this so that we can put this on our website afterwards. So if anybody does miss today, please pass it on to them. And thank you again for joining. So my name is Phil Crinan. I'm the director of the UKCRC Tissue Directory. But today I want to hand over to Emma Lawrence, who is going to be presenting on some of the work that we've been doing with COVID. So over to you, Emma. Thank you very much.
Thank you, Phil. Good morning, everybody. Thank you for joining us. Like Phil said, my name's Emma. I'm one of the engagement managers at the UKCRC Tissue Directory and Coordination Centre. I'm going to do a brief introduction about us and some work we've been doing, but then hand over to Lee Murphy, who'll be talking about some work they've been doing up at the Edinburgh Clinical Research Facility on COVID-19, and then there'll be plenty of time for Q&As at the end. So very briefly, just in case you're new to us, the UKCRC Tissue Directory and Coordination Centre, we were founded back in 2013 after the publication of this vision for human tissue. This was published by a consortium of UK medical research funders like MRC, Wellcome Trust, CRUK and a few others. And essentially they wanted to make better use of existing human tissue resources in the UK, and primarily to do this by making the existing resources more easily discoverable and accessible, and this was to minimise duplication of work within the UK. So we were established following this. We've since established the UK's National Register of Biosamples. So this is a map here of all of the places we have registered sample collections. We have over 200 different sample resources registered currently, and this covers anything from biobanks, but also things like clinical trial collections, cohort collections, and more smaller disease-specific collections. Research tissue banks must register as a condition of their favourable ethical opinion. We have a really strong data focus because obviously samples are a lot more valuable with really rich data sets associated with them. So we've established Europe's first information commons in partnership with industry. We do a lot of work to engage with different stakeholders. So for example, we've done some research on researchers, which we've published recently, but we also work with industry and obviously the biobanking community through events like this. We also represent the UK within a European network of biobanking infrastructure called BBMRI-ERIC. So I'm just going to talk briefly about a bit of work we've been doing to do with COVID-19 samples before handing over to Lee. So if you can cast your mind back to early March, we were talking about COVID-19 in terms of cases, not deaths. Unfortunately, things escalated pretty quickly. By the 17th of March, UCL, which is where I'm based, we're encouraging all staff to work from home. I know other universities had responded before that, so we became aware pretty quickly that biobanking capabilities were going to be impacted by these closures. On the 18th of March, we sent an email out to all 220 of our registered biobanks to find out how their capabilities were going to be impacted by the closures. On the 20th of March, that was when the full lockdown was announced. I know it's ages ago now. During this time, we were also advertising to researchers that we were going to be trying to help coordinating COVID samples within the UK. The 24th of March, we started sending out emails to any open biobanks with requests for samples from researchers. We sent a follow-up email on the 27th of March. On the 2nd of April, the first samples were sent between our first sort of biobank and researcher match. So that was just within 2 weeks of the first email that we sent out. So I think we were able to respond pretty quickly to the situation. And then on the 6th of May, we did a follow-up email. So essentially, I'm going to show you a bit of data based on these 3 mail-outs to our biobanks. These are the results of our mail-out. So out of the 220 biobanks we have registered, we had 81 that didn't respond. 17 were open with relatively normal activity. 33 had limited capabilities, so that you had a change of focus or less staff, and 89 were closed. So basically, out of all of the biobanks within the UK that we have registered, that's 40% of them were closed. So obviously this was going to have a big impact on the ability to collect samples for research within the UK. We tried to capture some of the reasons for the closures, but not everyone gave this information. And typically it was site closure or staff reallocation. So finally, just to share a couple of sort of success stories that we had from our work. So in total, we had 32 requests for samples from the beginning of the outbreak. We're still having some This is a lot more than we normally get, so we definitely noticed really high demand for these samples as expected. We were able to set up a collaboration. This is the first case study here on the left, between these organisations. So TAP-B is a biobank based in London at the Royal Free Hospital, and they were able to collect blood from COVID-19 patients, but they didn't have the facilities to process the blood because they didn't have any CAP3 labs. And they also didn't really have the capacity to deal with any of the logistics or contracts at the time. So we were able to partner them with Tissue Solutions, a company based in Scotland, and they were able to help with a lot of the logistics and the contracts. And also Cambridge Stem Cell Biobank, Cambridge, had CAP3 facilities and a bit of resource. They were able to help process the blood. So between the three of them, we were able to get the bloods out and blood products out to researchers that needed them. So that was a really great case study there. And then just another example here, Wales Cancer Bank were able to help. So we had some requests from researchers who wanted serum prior to January 2020 for use as control. And so Wales Cancer Bank were able to amend their ethics and release some residual serum as use as control serum. So these are examples, two examples of two biobanks who weren't actually able to collect COVID, but were still able to support COVID research. So it was really good to hear, to work with them, and thanks to them for their support. I think that's it from us. Just to say, if you can help, we have a form on our website if you're able to offer any resources, or if you have samples you can share, or anything in general that's gone well for you. It would be good to have a bit of a picture on behalf of the biobanking community so we can take forward some lessons learned. There's my contact details there, but we've also a chat box if you want to make any comments today. And I think now I can hand over to Lee. So Lee Murphy is Genetics Manager at Edinburgh Clinical Research Facility. He leads a team providing support to research studies investigating the role of genetics in health and disease. So over to Lee. Thank you, Lee.
Okay, thank you, Emma, and welcome to Edinburgh. I am in the lab today, so apologies for any external noises you might hear. So I'm going to talk about the work we've been doing during the pandemic to support research, looking at the different human responses to infection with the virus SARS-CoV-2. Next slide, please, Emma. Thank you. So I work at the Edinburgh Clinical Research Facility, which is a joint facility between the University of Edinburgh and the NHS Lothian. So we're a specialist facility for doing clinical research. We are MHRA phase 1 accredited for first-in-human drug trials in our clinical side, and then we have these different core areas listed on the right-hand side, things like imaging and mass spec. So we're trying to do clinical research in a multidisciplinary way, and I run the genetics lab here. Next slide. And so my lab is really supporting a whole range of genomics, particularly focused on clinical work. We do the sample processing, so that's lots of DNA and RNA extractions from kind of clinically relevant samples, things like blood, FFP. We do a lot of FFP work. We are also doing lots of sample storage within our freezers. We are not a tissue bank ourselves, although some of the projects we work with are tissue banks, things like Generation Scotland that we heard about last week. But we do have a lot of studies which aren't very visible, and I'm looking forward to working with TDCC further to see if we can make those more visible. And then we have our analysis side, so that's listed on the right-hand side, things like digital PCR, TAPMAN and genotyping assays, and we do lots of sequencing. So we're the main core facility for sequencing in the university, and especially for the medical school. Next slide, please. So we work on lots of studies. At the start of the pandemic, everything changed for us as it did for everybody else. Our clinical research facility, although it's a university building, it's embedded within the hospital site of the Western General. And this was then handed over to the NHS for clinical work. So our building became a haematology day patient ward for patients undergoing cancer treatments. And so this is our reception now. So kind of met with a nurse who takes the temperature and makes sure it's hand washing, and then sent upstairs to the clinical areas. and my lab's down in the basement. So, and so our facility changed. All our clinical staff then went to frontline work, mainly plugging gaps from people shielding within the NHS nurses. I guess for us in genetics, we mostly closed. We had a skeleton staff for about 3 weeks as we kind of changed all our research away from our normal research and just focused on COVID-19. Next slide, please. Okay, so last week, this tweet on the top right was sent out by NIHR, kind of celebrating that over 100,000 people had volunteered for a range of research projects. And on the left are those priority studies for hospitalized patients. My lab is involved in the top one and the bottom one, so 2 of the 5. But also in there, you can see the recovery trial. That's the tweet on the bottom right, which had their results out yesterday on dexamethasone, and great results there. So I think it's been fantastic the way clinical research has kind of refocused onto COVID-19, and kind of great results that have came out of it. But I think this is all obviously based on people volunteering for clinical research, and that has been fantastic to see as well. So I'll take you through the 2 trials that we're involved in. A plug for a recent trial just announced from Edinburgh University, and I thought I would plug this because I'm assuming you're quite a tech-savvy audience here. And so this is for people who have already got their genome genotyped. So maybe you've taken part in something like 23andMe or Ancestry, if you're interested in genealogy. And if you've got those genotypes, you can upload them to this site, the Coronagene site. And this allows the investigators to do what's called a genome-wide association study to look at which bits of the genome is involved in COVID-19 infection. So it might be something you want to think about. So I just put that up there as a wee plug. So next slide. Okay, so I'll go through the 2 studies that we're involved in. So the first one is called Genomic, which stands for the Genetics of Mortality in Critical Care. And this is run from Edinburgh University by Kenny Bailey, who's a clinician that we've worked for many years. And it's a project which has been recruiting since 2016. And it was really set up for future pandemics. So things like, the next flu pandemic and such like. So it was, the groundwork was already done, and then it was able to just really ramp up as the COVID-19 situation got worse. And so far they have over 200 ICUs across the country, which is about 5,000 intensive care beds who are recruiting for this study. And so it's fairly simple from a patient point of view. It's just one blood sample, EDTA blood sample, which is sent to my lab from across the UK. But the data is then linked all through REDCap. My lab gets the blood tube in, we do the DNA extraction, then we're genotyping in the lab using the Illumina Global Screening Array, which is looking at about 730,000 variants across the genome, so that we can try and look at which variants are involved in people having worse outcomes or those who are having better outcomes. And then as part of this, we're sending an aliquot of our DNA down to Genomics England, down in Cambridge, so that Illumina can do the whole genome sequencing. Okay, next slide, please. So the next study then is called ISARIC, which stands for International Severe Acute Respiratory Infection Consortium. And so this is an umbrella organization. Genomic is part of this as well. This particular study is the 4C part of it, which stands for the Coronavirus Clinical Characterization Consortium. ISARIC is kind of an umbrella organization, and it's all about putting protocols in place in advance of a pandemic. Again, this is all work that's been carried out before COVID-19 started. And it's really about sharing all these data resources. There's a very big ethos of sharing within ISARIC, 4C and Genomic. And this study is really kind of trying to look at the basics of the infection, who's getting infected, how and why, and what are the kind of comorbidities involved. It's got this very pragmatic methodology where the tier 0 is essentially just data collection. So you get the data from the patient and you input it onto a database which is already kind of been made And so everything's in the same protocol. And they just passed 60,000 patients recruited last week on Friday. As part of this, then there are tier 1 and tier 2 sampling. So if you've got the time and the ability, you can collect a single sample from a patient, a single time point sample. And if you've got more ability, you can get serial samples, so on days 1, 3, 9, and 28. And the idea is that, you know, if your ICU is not so busy, you could get more samples, but as you get busier, you would drop back down to tier 1 or tier 0. So it's very pragmatic. It's also run by Kenny Bailey locally, and also Callum Semple in Liverpool and Peter Shaw in Imperial. Next slide, please. Okay, so data that's come out of the clinical characterization protocol was published last month in BMJ. This is a project led by Anne-Marie Doherty here in Edinburgh. And so looking at the first 20,000 patients, it really kind of gives you an overview of what the disease is looking like in patients. So the top left, showing that it's essentially a disease of older people. The bottom left is that it's predominantly in males compared to females. The middle slide, the left side, is showing those kind of symptoms that you see with COVID-19. So that's the cough and the fever. And then also that kind of gives you data about, you know, who should be self-isolating, what symptoms to look out for. And then there's the comorbidities, so that's giving information on who should be shielding. So the chronic cardiac disease and diabetes, those kind of things. So this is all the data taken from the first 20,000 patients. It's now up to 60,000, and every week this data is downloaded and put onto the ISRIC 4C webpage, which is on the bottom right. And so you can go and look at this data in real time. And if you wanted, you could put in your details, your gender, your BMI, if you've got any comorbidities, and you can look at your risk of hospital outcomes from COVID-19, if you so wish. Okay, next slide, please. Okay, in terms of sampling, so I've mentioned this single samples or serial samples. Each time they're getting a huge range of samples, as you can see here, serum, plasma. All these sample types have been collected and sent to the main labs in Liverpool and Glasgow. They're BSL-3 labs, and then they take the samples and they send them out to all the different labs who've got specialities in different areas and who are then using them for research. And my lab is getting the two bottom ones, which are essentially the cell pellets or the blood samples, which we use for DNA extractions, and also blood for RNA extractions. So this is a huge amount of work for the labs in Liverpool and Glasgow. And they've done a fantastic job in receiving and sending these out. And the quote on the bottom right, then I've added, because I thought it'd be quite good for this audience. It says, "We will share samples to get answers as fast as possible. We will not end this outbreak sitting on a biobank. Our intention is to use every drop of every sample to have the biggest possible impact on the COVID-19 crisis," which I think is a very strong statement of intent. Okay, next slide, please. Okay, so for ISRIC 4C, for the DNA extractions, we're doing exactly the same as genomic, doing the extractions, doing the GWAS on the Illumina GSA array, and sending an aliquot to Genomics England. The difference with this study then is that we're getting the RNA, we're doing the extractions, and then we're doing messenger RNA sequencing on our NextSeq 550 or 2000. And for those who are interested, the method is here. We're using the NebNext Ultra kit. Okay, next slide, please. Okay, so why are we doing this? So I guess there are lots of variability in outcomes of people who've infected with SARS-CoV-2. And some of that will be down to genetics. And the idea of the genomic and ISRIC is to find those genetic influences and variants. And so this is something we've done before during the H1N1 pandemic a few years ago. We worked with Kenny and his team. We did a GWAS on those samples, and we were able to identify IFI-TM3 gene as a risk factor. And there was some nice functional work which took place for this Nature paper. And then just recently on Medical Archive, although I think it's now published in New England Journal of Medicine, is this GWAS for COVID-19, which was showing hits in chromosome 3 and the ABO blood group. So I think we will see if our GWAS finds the same and replicates this. Okay, next slide, please. Okay, so how did we do this? So I think we were in a very good position at the start of the pandemic to help out in this kind of research. As you saw, we have this kind of pipeline going from patients through to the analysis. So I think we were in quite a strong place, but it did lead to quite a lot of challenges to overcome. There's some opportunities as well, but quite a few challenges. And I think the biggest right at the start was getting the risk assessment done. So, okay, so once we got the risk assessment in place, then the next biggest worry, the thing that kept me awake at night, was how we were going to do this amount of work. So I would say this is a graph of our DNA extractions that we completed through the years. Generally, it's between 3,000 and 4,000 DNA extractions, about 1,000 RNA extractions. So we're quite a small to medium lab. But for genomic, we were talking about doing 10,000 samples in 6 months. So that's quite a step up from what we were doing previously. Next slide. On top of this, then there was the limitations put on us by the COVID-19, trying to do physical separation particularly. So as part of the risk assessment, all the work had to take part within our Cat2 lab. We got sign off for Cat2. We couldn't have done it within a Cat3. And we had to do it in our safety cabinets, especially anything that caused an aerosol. And so that's loading and unloading the centrifuge buckets particularly. So we have those hoods in our lab, but that's a bottleneck then. On the left is our big hood. That's a big 2-meter hood. And usually technicians like working in there because they can work side by side and have a chat while they're working. But we had to halve this capacity to keep the 2 meters apart. And on the right-hand side, we had 2 separate hoods there. That's pictures taken before the pandemic. We've now separated those, so there's 2-meter distance between them. So that was a real bottleneck in the lab. And then also not having everybody in the team in the lab at the same time. So we've reduced our staffing down, so nobody's working more than 3 days a week. And again, to help with that kind of separation. So we were having to increase our throughput with less staff and kind of less resources. Okay, next slide, please. There were opportunities as well. So it allowed release of some extra money so that we could buy new equipment. So one of the things we bought was the PerkinElmer Chemagic 360, which is an automated DNA extraction device. Unfortunately, I think just about everybody in the UK was trying to get one of these. So even though we ordered it about 2 months ago, it's still not here. And I don't think I've been particularly happy with PerkinElmer as a company, but we'll see what it's like when this arrives in the lab, and hopefully this will increase our capacity as well. We also bought a Cubic Flex to increase our qubit QC, mainly for our sequencing libraries. We've got the Mantis, which is the green little robotic platform for doing miniaturization and speeding up our making up of QC plates. And then on the right-hand side, we've got our new sequencing machine, the Illumina NextSeq 2000, which we were in the process of buying before the pandemic hit, but which we managed to continue and get delivered during the pandemic, so we could continue with our RNA sequencing. And it's a new platform, and just because most labs were closing, we're now one of the very few labs in the world to have this platform, and that's been a great boost to us and increased our profile within Illumina, the company. Okay, next slide, please. Okay, and finally, so we've been able to make some new links out of this as well, which is always exciting with the TDCC, and I'm looking forward to pushing that forward, seeing how we can work with them. Genomics England, we hadn't worked with them before, and that's been quite an experience, mostly enjoyable. But most important was working with NECWAS, taking part in their DNA quantification study. So this is a study where you get given some DNA of known concentration, you can put it through your protocols, you can compare yourself to others. And in this case, we were comparing ourselves with the Illumina labs who were going to accept our samples for whole genome sequencing. And it was a way of us being able to, I guess, benchmark ourselves and kind of check out the quality. And it's something that we've been considering before, but had never actually taken part. And this really pushed us into doing that. And thanks to Sandy Deans for helping us with that, and also coming to do an audit for the lab on behalf of Genomics England. Okay, next slide, please. Okay, so how have we been doing? So it's been a very busy period, and it feels like we're kind of coming out of that busy period now, which is quite a relief. So this is data on the graph from the genomic study, just genomic here. So we had about 300 samples in the lab already for this study before COVID-19. We had about 3 weeks where the lab was closed, or at least we just had a skeleton crew on as we still had samples, clinical samples being delivered and just putting them into freezers. And then the samples really took off then from the middle of April. And huge numbers of samples come into the lab, which is the blue line. The orange line is the number we extracted. And we've really kept a track of the number of samples coming in. We managed to extract at the same rate, which I was worried about that we were going to get swamped, but actually the guys in the lab have done a fantastic job there. And the grey line is how many were in storage and that we still had to extract. And I was keeping a track of this because I was really worried we were going to run out of freezer space for the blood tubes. And you can see from the angle of the graph, the pandemic is kind of flattening out, which is great news. Although we were hoping for 10,000 in 6 months, I don't think we're going to get there from the serious cases, but as we kind of start taking on cases which were maybe not quite as serious, we'll increase those numbers again. So at the end of this, we've extracted more than 3,000 DNA samples in a very short period of time. We've genotyped over 2,000 of them now. We've sent about 500 down to Illumina for sequencing via gel, and the plan is to get that up to 1,000 by the end of this month. And then by the end of July, there should be 1,000 whole genomes in the research environment for people to look at. And then for the RNA, we've extracted 500 samples, and we've sequenced over 200 now. So I think, yeah, I think the guys in the lab have done a great job here. Okay, next slide, please. So I think we've achieved a lot over the last 12 weeks. I think it's been quite a positive experience. I've really enjoyed just focusing on these 2 projects. Usually we're working on 60 projects at a time. It's been quite good just being able to focus on one thing. I've been very impressed by my team. Although I already knew that they were a great team, it was very satisfying to have that proven how flexible and hardworking they've been through this. And I think we've all found it very rewarding being able to contribute to COVID-19 research. And then for me personally, I'm involved within the university, what's called the Technician Commitment, which is a UK-wide initiative to raise the profile of technicians. And I think within Edinburgh University, and I think within the sector as a whole, it's been very satisfying to see the hard work of technicians. Generally, they've been the last to leave labs during the close down and been the first back in to reset them up. And I think the pandemic has really helped raise the profile of technicians and the fantastic work they do. And similarly with core facilities as well, like mine, that when we come back, we're not going to be at full capacity within the university. And researchers are going to really rely on core facilities to get their research done. And I think that's going to be the next challenge for my lab, is how do we continue with this COVID-19 work, but still help those researchers we've been helping previously and get their research back up and running. Okay, next slide, please. Okay, so we're at the end. So these are, massive projects, loads of people involved. I'd really like to thank Kenny Bailey, especially for believing in the lab, and he's a big supporter of ours. Locally within Edinburgh, Andy and Wilner have been a great assistance in helping us get up and running. Andy with the data, and Wilner essentially getting the risk assessments through the university, which was a fantastic job. There are huge labs within Liverpool and Glasgow that I mentioned, working in very difficult conditions. And particularly want to thank Hayley and Shona in Liverpool, who are my main contacts there, and Sarah in Glasgow, Sarah McDonald. Next slide, please. Mostly I'd like to thank my team. They've done a fantastic job. I'd like to particularly thank Kirsty Morris, who's at the back on the right with glasses on, who's the lead technician for the genomic project, and then Katrina Fazi, who's in the front middle in the white jumper, who's the lead technician for the ISERIC project. And then a quick word about Lorna Donnelly on the right hand side, who is our new technician, and she just started 3 weeks before shutdown. And so big thanks to her as well, and she's been able to contribute as well. Okay, thank you very much, and happy to take any questions. I think Phil is, I'm handing over to Phil now, I think.
Yes, thank you very much. That was really, really interesting. So we've had some questions come in. So for everybody, there's a Q&A button at the bottom if you've got some questions to ping it across. So we've already had quite a few come in, which is really good. So I think the first one, so two people have asked this question around social distancing. So how will this affect you, Lee, in terms of the change or the potential change from 2 meters to 1 meter? Will it make much difference for you?
Yeah, so I mean, I guess that's going to be English policy and not Scottish policy. I think we'll be a wee bit behind there. But yeah, as soon as we can, as soon as that reduces, you can get more people in the lab, essentially. I don't think we're always going to have that bottleneck with the hoods. We'll never get 2 people in our big hut, so we're limited to 3 huts. The biggest change is when we get the Chemagic system in. That'll help us. Once you've got past one bottleneck, you just come across the next bottleneck, which is QCing the samples and getting the qubit done. Once it reduces, then we can get more people in more areas of the lab. So it just helps everything, really. We can probably increase from 3 days a week upwards.
Brilliant, thank you. And so a comment's come in as well about how does ISERIC foresee in genomics share data with global researchers?
Yes, good question. So it's as soon as the RNA data is going on onto what is it called now? ENAQ. So this is the database where anybody can then download the reads. So everything's been made as widely available as possible. So yeah, I mean, that is part of the ISERIC protocol, that you have to make the data as available as possible to researchers.
Yeah, that's perfect. I think there's some work we can do together as well on making it more visible to people that may not be aware.
And yeah, and all those sample types I mentioned today are all available on request. So the team are obviously working under very difficult circumstances and are getting things out to the research groups who are specialising in different areas. If you think you can add something over and above that, you can request those samples and they can be added to the list of samples that get sent out. And that would be Kenny or Callum or PT who would contact for that.
Perfect, thank you. So I'm going to keep firing them at you while they keep coming in.
That's
so
okay.
Does Forsey have a feel for the reliability of home-based testing, for example, BP and BMI versus clinical-based?
Oh, right, and it is telemedicine and remote sampling collection in the future, right? So, yeah, I don't know the answer to that. So it's not specifically set up to test the reliability of home-based testing kits. And it's not, the tool that's on the web is not a clinical tool for diagnostics. It's just there to give you a feel of the outbreak. And so I think there's a difference in there between pooled data samples and specific diagnostics for an individual and they're not doing that.
Okay, thank you. So we'll take the one just on the external factors. So travel to work, how I mean, all those things that everybody has to cope with. And I think just to pick up on your comment around the commitment to technicians, I think it's definitely something that's come through so strongly in this current situation. So it's obviously a very good national effort that's been going on around recognition of technicians, but I think this really demonstrates their value. So it's brilliant that you also demonstrated that within your presentation. But how did they also cope with those types of external factors? Did that have much of an impact on you in terms of travel to work, kids at home?
Yeah, so I mean, travel to work is interesting. More people are cycling, which is always a good thing to see. And the council has just put new cycle lanes out by the hospital. I always cycle anyway, or run in, so I'm pleased to support that. More people in my team are cycling. For those who live further away, it was actually a really easy trip in, because they could park right up at the hospital, which you could never usually do. But that's stopping now as people are coming back to work. I guess the family situation for kids and such like is just something that I have to deal with as a manager, that we all have to, well, we all have that going on in our life. So, you know, I can only work 3 days because for 2 days a week I'm homeschooling, same for other people in my team. And so we're just trying to work our way around that. I think what I've been surprised about, or one take-home message from this, is how much work the technicians could do from home. And I wouldn't have thought that before. I thought they would have to be here contributing within the lab, but there's huge amounts they can do from home. And, you know, at the start of this, when we were just trying to get things started, it was all mainly about CPD and getting our SOPs refreshed. And then at the end of it, we're doing lots of the kind of paperwork things at home. And I hope that we keep on doing that. That was a big lesson for me. But the other thing then was we don't have anybody shielding, but we did have a few people self-isolating. And that was near the start when everything was getting really busy, and trying to cope with that from week to week was quite difficult. And trying to build that into the working rota.
Yeah, and a comment came in about the very impressive scale up as well that you managed to achieve. As you talked about the worries that you're going to receive all these samples, not be able to process them. So being able to ramp up like that in those circumstances must have been a real big achievement there.
Yeah, I'm very proud of the team for doing that. And of course, there's lots of knock-on effects, you know. So trying to get deliveries in has been difficult. Our autoclave is 25 years old and a wee bit on the blink, so getting engineers in was difficult. And so, you know, there's lots of people that you rely on. We were one of the few labs back, but our sister lab at the IGMM just down the road, at the end of the corridor, they were all away, and they'd be our first port of call for help, you know, for support. And they were all away. So things like our lab coats are all washed through the IGMM and things. So it did, there were lots of knock-on effects, which I hadn't really considered when I first said, yes, I'll do this, Kenny. I'm happy to do it. Yeah.
Yes, it definitely has made people have to adapt.
It shows what a big teamwork it is, I think. You know, it's not just my team, there's a big wider team out there, I think, which I rely on. And it's kind of brought that home to me, how much I rely on other people.
Yes, definitely. So we'll pick up just, I think, maybe just one last question. So it's from Joe Butler. So given that you don't want to leave any stone unturned with regard to COVID research, is there interest to adopt long-range sequencing in future and optical mapping to look for larger events?
That's a good question, Joe. There isn't at the moment, and that's mainly due to the technique we're using for our DNA extractions. Optical mapping then isn't an option. And these protocols are put in place a long time before the pandemic. So I think there are other research groups who are better placed to do that work than we are. Okay.
Well, we two more have just come in. So it's around the question around ethnic differences, but then also around data sets. So how how it's possible to link data sets, or potentially if they're anonymized or not, and how that can occur. So first one was ethnic differences. Have you seen anything?
Yeah, so that's the question I can answer the easiest, because the ISERIC group just last week released the paper on the effect on BAME communities, showing an effect for diabetes, and so that is on the archive if you want to look for that. That's Anne-Marie Doherty again. So you can look at that paper. So they have been able to tease out those ethnic differences. And the genotype in particular will be able to do that as well, because of the array we've chosen is a global screening array. So it does have genotypes from across different population types. It's not biased for Caucasian as a lot of other arrays are. And then mixing data sets. So yeah, I mean, that's probably outside, my area of expertise, but I know, for example, the recovery trial and ISERIC are working closely together. And so all these different trials are looking to link up their data sets. And obviously if you're going through REDCap and things like that, then you can link that data. But it's not an area that I'm an expert in.
Okay. But thank you for picking up the questions, and thank you very much for your presentation today. It was really fascinating, and thank you, Emma, as well, for showing the work that the tissue directory was doing in trying to coordinate some access to samples when we had so many requests come in. And thank you, everybody, for attending. We did record today, so we'll be making it available on the website shortly. And we do have a final webinar coming up very soon. The date's still to be confirmed, but it'll be around some of the database systems that you can utilize within your biobank. So that's one to come soon, and we'll be in touch around the date for that. So thank you very much for attending. Have a good day, and hopefully see you soon.